Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. First, we'd like to thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Last month, we launched the Dr. GPCR Symposia event series. Our first symposium was on March 24th, 2023, and it was on challenging GPCRs. It was a day filled with amazing talks, great discussions, and we hope the beginning of new collaborations. In case you missed it, you can watch the talks in the Dr. GPCR ecosystem with your premium membership. Also, mark your calendars for the next symposium coming up on May 19th, during which we will hear from speakers on GPCR activation and signaling. At all our symposia events, including the one coming in May, all trainees are welcome to present a poster. There won't be any poster selections, and everyone is welcome to join us for the poster/networking time. For more information and an updated schedule can be found in the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You can join us live by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. To navigate the ecosystem again, please use the direct links in the footer. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Dr. Kenneth Jacobson as my guest today. Hi, Ken.
Hi.
It's
Hello,
so nice to
everyone.
meet you.
Nice to meet you as well.
I forgot to ask, do you prefer Ken or Kenneth?
Ken, please.
Perfect. Because we've exchanged a couple of emails in a different setting, and I always like to ask these questions. I'm so happy to have you on. So why don't we start at the beginning? Would you please introduce yourself and tell us where you're located right now?
Okay, I'm a researcher in the NIH intramural program. You know, there's the extramural that gives out grants. I'm actually work on site in Bethesda, Maryland, in the National Institute of Diabetes and Digestive and Kidney Diseases. My lab is called the Molecular Recognition Section, and we're in the Laboratory of Bioorganic Chemistry. And we work at the interface of chemistry and biology. I'm trained as a chemist, which I'll get into a bit later. But when I first started my group, I made sure to have a biological component. So we have both experts in biology and the chemistry in our group.
Which is great because I think it's so complementary. And sometimes, although it's sometimes difficult to have conversations between biologists and chemists, being able to be in the same group and learning the same language, or to use a language that crosses the disciplines, is really important. So you mentioned that you trained as a chemist. Did you always know that you wanted to be a scientist, or was there a time in your life when you were like, okay, this is what I really want to do?
I would say I can't remember a time when I didn't think I'm going to be a scientist someday. I was actually inspired to take an interest in chemistry by my grandfather. So this is going back. He was the youngest of 10 children that immigrated. The whole family immigrated from Latvia. They were all business people. He was the first one to eventually go to college, and he graduated in 1914 with a degree in chemical engineering from University of Michigan, and all of his siblings pitched in to support his college education.
Wow.
But by the time I came around, he had already accomplished a lot in his field, and he had inventions. He had his own little company in Cleveland, Ohio, and I used to go to his lab on weekends and just poke around, getting to know the glassware and some of the chemicals and familiarizing myself at least with the names and some of their properties. Can you imagine letting a 9-year-old child roam an organic chemistry lab by themselves without any immediate supervision? I wouldn't do that to my children, but I didn't get into any trouble. I was
That's
well behaved in the lab.
that's great, and that's important. I don't think that that is something that could happen nowadays with all the rules and regulations.
Yeah,
But I think it's really inspiring to be able to be in a lab at that young age and
yeah,
being able to see
yeah.
all the lab setting and the
Yeah.
glassware.
Plus the concept that everything you see around you is made up of less than microscopic atoms and molecules. And I got that very early on.
How great. Did you ever get a chance to work later on in the lab with your grandfather?
Not actually working in the lab, but I would ask him questions, and he would explain chemical bonding to me on the rudimentary level. And then I took home some of the chemicals from his lab, and I started my own little lab in the basement. And I was known in school as being interested in chemistry, and I was selling some of the chemicals to my friends who wanted to get interested too. And when I was in fourth grade, I made soap and
Yeah.
sold a little sphere of soap to my teacher, and that was uh, all those experiences contributed to my eventual entry into chemistry as a profession. When I was 12, I wrote an autobiography that I'm going to be a PhD chemist, and it came to pass.
How amazing is that? That is amazing. You mentioned soap. I also make cold processed soap, and now I switched to liquid soap. And I have to say that, you know, for a teenager back in the day without the internet being available, making soap, it's not difficult, but still with today's tools, you can go on YouTube, you can learn how to do those things. But kudos to you for making the soap that young. So where do GPCRs come into the picture in your track? You
Yeah. So my higher education, proceeded at Reed College in Portland, Oregon, which is a high-level academic school. They only have undergraduate students, so you were taking all your courses in sections with actual professors, and that was also very inspirational. And you have to do a senior thesis, and in the sciences, that means an original laboratory project. So I started working on enzymes and attaching chains to enzyme ligands. And it actually fed in conceptually to what I did in graduate school, which was in the laboratory of Murray Goodman at the University of California, San Diego, working on peptides. I was trying to couple beta adrenergic agonists like isoproterenol to peptide carriers in a way that would retain the biological activity. And that actually led to my own project when I got to NIH, because I wanted to apply a similar strategy to a different receptor. And remember, at that time, and this was the early 80s, I came to NIH in 1983. So at that time, nobody had any idea what a receptor was chemically, possibly a protein, but not necessarily. So the beta adrenergic receptor was cloned in 1986, and it came as a surprise that it had the same overall motif as rhodopsin. And everybody just assumes, well, didn't we always know that? No. So when I got to NIH, I was looking for a receptor test system to apply this strategy, which I called functionalized congeners, where I would take a pharmacophore like caffeine and attach chains at different positions and see which position was insensitive in the receptor binding, and then build out the chain in a way that would not only preserve the affinity and selectivity, but somehow enhance it. So I was thinking in terms of using the strategy to make drugs. to actual drug discovery with things with chains on them. And, as that is very obvious now, a valid approach. But at the time, it really ran against the conventional thinking. And I was told by a senior chemist at NIH that I didn't have enough respect for the molecules.
We're not respecting the chemistry.
Right, because in those days, the drug was a very compact molecule. And if you put a chain on, the idea was you can't get much beyond ethyl or propyl. So I really had to fight against that perception. But finally, my positive results won out, and I was granted tenure after about 6 years at NIH.
Wow, that's amazing. And it's interesting, but as you mentioned, it is obvious now that having a scaffold and adding branches, decorating those branches might result. Well, it's interesting to study what is the effect on a receptor, and can we make a ligand bind tighter or increase the affinity or even change the signaling. But I can only imagine at that time in the 80s where we didn't have structures. We barely were at the beginning of in the late 80s, early 90s, the beginning of the cloning boom. So it's interesting that at that time you already had these ideas, which obviously were proven by the data, because at the end of the day, as scientists, we can argue up and down the corridor. The data is the data.
Yes, and I was dreaming of structures. I figured before long we would have structures. It took a long time, but and I was always thinking about these chains I was attaching, and thinking about how that would affect the surface of a hypothetical receptor that was embedded in the membrane.
Right.
So I was well prepared when the structure actually came out, and it's nice to show a little diagram that I drew of one of my adenosine antagonists, and then the actual structure of that very same antagonist in an X-ray structure. It's almost one is the cartoon of the actual reality. So I was on the right track.
That's great. Did you celebrate since you mentioned that the cartoon resembled very much the actual structure that was published, how did you feel about that when the structure came out?
Terrific. It was one of the high points emotionally. And, as scientists, we don't always have high points, but this was certainly good feeling.
Agreed. I think I say this enough times now. I said this enough times during the podcast. I feel like as scientists, we go from dopamine rush to dopamine rush. And it's that rush the wave that carries us to the next one. And sometimes the distance between the two peaks of dopamine, when something really works out, is pretty long. So we do have to build that thick skin.
Yeah.
To go for it.
So one thing I would like to add is, how did I get to adenosine receptors? So when I first arrived at NIH and I was looking for a receptor, just by coincidence, I was across the hall from Dr. John W. Daly, who was a famous natural products scientist. He was the one who was most famous for collecting poison frogs and figuring out the toxin principles of toxicity of the poisoned dart frogs. Anyway, he immediately suggested adenosine, and I have to admit I had never heard of adenosine receptors because it was very obscure. There were only a handful of people in the world even thinking about adenosine receptors. Yeah. So this was a very good choice, because I got in at the ground of a new field that greatly expanded and is still expanding.
Yeah. Yeah. And especially with, when I think about adenosine, I think about caffeine and its effects, but there is the receptor itself has so many functions, and it's also implicated in cancer, for example. So it's a great system to study, which leads me to my next question, what is your favorite GPCR?
Well, I just have to say families of GPCRs, and they're the ones that I've been studying for a long time. the four adenosine receptors. And among those four, I would say the A3 receptor is my favorite. There are also the P2Y receptors, and that field has lagged behind the adenosine receptors for a number of reasons. One reason is that people doubted the existence of signaling properties of nucleotides outside the cell. And the champion of that concept was Professor Jeff Bernstock from University College London. And it took him about 20 years to get people to take him seriously. And in fact, he was vindicated with the cloning of the first P2Y receptor and later the P2X ion channels. And I got to know him from an international conference in 1987. And I told him even at that time, I wanted to collaborate with him. And this was before people really took him seriously.
Yeah.
But he was thrilled that a chemist got interested because he knew how important it is to have chemical tools to define pharmacological actions. And he had a deficiency of pharmacological tools at that time. So I was very excited and took a few years to get started on the P2Y receptors, but we eventually struck up our collaboration and made a lot of progress.
So you mentioned we can come back to the P2Y receptor a little bit later on. You mentioned the four adenosine receptors. If you were to tell us a little bit more about the differences between these receptors, that would be really, I think, really useful. I don't know enough about these receptors. What are the main differences between them, and why are these receptors important?
Well, two of them are coupled to GI and two are coupled to GS. The A3 receptor is unusual in that it doesn't have any severe cardiovascular effects. So there have been many efforts in the past to conduct clinical trials and eventually commercialize agonists of the A1 and the A2A receptors, and a little bit dealing with the A2B receptor. A2A and A2B are both GS coupled. Yeah. A1 and A3 are GI coupled. Anyway, many of those clinical trials got derailed because of these cardiovascular side effects. And the A3 receptor seemed like a good choice to me because, well, number one, it didn't have these predicted effects, but we were also the leaders in developing ligands for the A3 receptor from the very beginning. So we had tool compounds that could then be modified and optimized chemically. And then so it was a question of what were they good for therapeutically. So I'd like to add that when I first start working on a receptor, it is an intellectual challenge. Here's a new receptor. Can I make an agonist, an antagonist, or some other modulator? And that's what we did with the A3. And I have to say, we even beat out Merck, who was on the trail of A3 antagonists, and we published before they did. So I'm happy that our work led to that. Actually, so to tell you a little more about the A3 antagonists, yeah. Caffeine, which is very good at the other adenosine receptors, is poor at the rat A3 receptor. And that's all we had to work with initially. So this was in the mid-90s, before they had chemical libraries that you could just buy.
Yeah.
So I tried to assemble my own chemical library. So I went around NIH and asked various chemists, and there aren't too many chemists at NIH, let me tell you. But anyway, I got boxes of compounds from people who said they didn't need those anymore. And I thought, I looked through each one and thought, oh, okay, that might be a good one. And we found 5 or 6 hits from that library of a couple thousand compounds that I assembled manually. And then we optimized those compounds and increased the A3 affinity and decreased any other activities that were known for those compounds. So that was a successful campaign. But at the time, we thought A3 antagonists would be more useful therapeutically, but it turns out the agonists are more useful. And we were on the trail of agonists in collaboration with Gary Stiles at Duke University, and we developed the right tools. And over the years, we've gotten to selectivities of 10,000-fold or greater in binding to the A3 receptor. So we don't have any off-target other adenosine receptor effects. So that's worked out well.
Well, that's great. And these things are really challenging, especially when you have multiple receptors of the same family that have similar, they're activated through the same G protein, or they have similar signaling effects. From a therapeutic standpoint, what kind of diseases are these in general adenosine receptors involved in, and why was it more important to have an agonist for the A3 instead of an antagonist?
Well, let me start with the last question. In the mouse, the A3 receptor causes release of histamine, and people thought, well, if you can block that, you might have something for asthma, right? Mm-hmm. Yeah. But the mouse biology is not equivalent to the human biology. And it turns out that in vivo, people who tried to use A3 antagonists in rodent models, they didn't work real well. Okay. And also for cancer models, there was a school of thought that you need an A3 antagonist for treating cancer. So in vivo, that was the real test of these compounds. The agonists had an anti-inflammatory effect and a growth suppression effect on tumors in, for example, nude mice models of human tumors. So the proof was in the in vivo tests. As far as what all the adenosine receptors are good for, that's a big question. We don't have time, but the short version. One of the hottest areas is blocking the A2A or A2B receptor. You probably are aware of this, but in cancer immunotherapy, you want to suppress the anti-inflammatory or immune suppressive response of adenosine on a variety of immune cells. So you can either administer an A2A or A2B or mixed antagonist, or prevent the generation of adenosine with various enzyme inhibitors. And all those versions are being tested now in the clinic, and it's looking very encouraging. A2A antagonists are also cerebroprotective, so they could be good for Alzheimer's disease, for example. There's already one drug on the market, which is a co-therapy for Parkinson's disease, which relieves the symptoms of Parkinson's disease, but it also in the long term, it might be proven cerebroprotective, disease-modifying compound. So there's a lot of hope for chronic disease treatment for various adenosine receptors.
I think it's fascinating. Yeah, I was aware of the A2A trials and in the context of cancer and the need to inhibit the receptor. Let's go back a little bit to the P2Y. Where does the P2Y program fit in your work?
Well, P2 receptors, both the P2Y GPCRs and the P2X ion channels, are pro-inflammatory in general. Mm-hmm. So we've focused on making antagonists, although we did a lot of work on agonists, but now that's kind of passed for us, and we're focusing on antagonists, particularly of two subtypes, the P2Y14 and the P2Y6 receptors. P2Y6 is lacking effective antagonists. We introduced one, which acts as an irreversibly binding compound, and actually leads to protein degradation of this receptor. You might not be aware of it, but this compound, which is a diisothiocyanate, therefore chemically reactive, reacts with a particular cysteine on the intracellular loops of the P2Y6 receptor. This was just published about a year ago, and that goes into the proteasome system, and the receptor goes away. So that's even better than an antagonist. But that's not really a druggable compound because it's chemically reactive and not very stable in aqueous medium. But it's good. It's a useful tool compound. Anyway, we would like to get competitive antagonists for studies of in vitro and in vivo pharmacology. And we're working. We're not there yet. The P2Y14 receptor, we've done a lot of SAR in that area, and we have some naphthalene derivatives, for example, based on a patent from Merck, but we've greatly expanded the SAR. Mm-hmm. I should mention that my staff is mostly at the postdoctoral level. So we have very talented chemists who come in and just do a great job of generating new compounds. But we don't just make compounds randomly. We always have close communication between the chemists and feedback from the pharmacologists in our group. So we don't waste time making a lot of compounds that aren't going to get us anyplace. And with this very focused approach, we've been able to optimize both the affinity and the physical chemical properties. So we have some compounds that might actually be suitable as pharmaceutical agents. I should also mention that some of our work is in collaboration with small companies. You might wonder, how does the government work with companies?
That was my next question. Yes.
Okay. So it has to be through a very well-defined framework, which is called CRADA, mm-hmm. the Collaborative Research and Development Agreement. And this guarantees the rights for the company to license the technology without having to jump through a lot of hoops that somebody off the street would have to jump through. So it's really beneficial. And we've had some very successful CRADAs. And we have 4 drugs in clinical trials already, but I can get to that in another question. Anyway, back to the P2Y14 receptor. I've had a whole series of postdocs who have done beautiful jobs at generating useful analogs of various sorts with fluorescent labels. We actually use a high affinity fluorescent antagonist now for our bioassay. There's no radioligand for that particular receptor. We've tried. We tritiated one of the compounds and it sticks to everything. All plastic. It just sucks it out of solution.
That's amazing. It sounds to me that you have a very well-defined set of programs with a very well-defined pipeline in the lab, and you've built a team around a well-established team of more senior researchers to get these programs moving. Here's my question. It's how come you stayed in academia / government and not moved into industry? Because the way you're talking about these programs and the way you're describing the research, it sounds to me that it's very industry-like.
Yes, well, I think that's a correct perception. Of course, on a tiny scale, I don't claim that we can compete with the development programs in industry, but an academic collaborator once called my group Merck and Microcosm. Somebody else had that idea. Uh, I've always been curiosity driven, and that's what really fuels my excitement about science. And that I can do that at NIH because uh, we don't have to write grant proposals. That might come as a shock to many people, but being in the intramural program, we only have to justify our progress after the fact. So I have the ability to be more creative and exploratory. And I don't have to ask the management if I want to follow up on some interesting finding. So this is really my comfort zone. And I've had some feelers from companies. I've visited a lot of companies at various times. And I have high regard for their research operations, but my own desire is to stay here, even though it probably would have been more lucrative to go with a company. I've never been motivated in any huge degree by financial considerations. When I took the job at NIH, I had another option, another offer for a lot more money at a university, but I thought that NIH would give me the right opportunity to grow my program.
I think,
Turned
I
out
think not a lot of people know the benefits of the intramural program at NIH, which is truly a great thing to have.
Yes, and in fact, if you look at the history of accomplishments of the intramural program, you see that it's been very important in the beginnings of cancer chemotherapy and just, a huge list of accomplishments. So whenever I get into discussions with people about government funding, and you can imagine not everybody wants to see more money to research funded by the government. I try to defend strongly, and I believe strongly that the research operations like the NIH and intramural program will pay for themselves in the long run.
Yeah, no, agreed. How did you, because obviously being in an intramural program and just coming to NIH is definitely not enough to set up a micro Merck operation like yours. What was the thought process behind putting together your team the way it is now? And my best guess is that it evolved with time, but
yeah,
I'm really interested in that. I think this might be interesting for junior scientists.
Yes. It took a few years before I was invited to get onto a tenure track program, and that was through publication. And I really took initiative. I was proactive at designing new concepts that I could explore experimentally. And my mentors at NIH, both my immediate supervisor and my collaborator, John Daly, were very supportive. And that's an important point. Get a good mentor in your career. At any stage, you can use a mentor. And that has to be somebody who cares about your future. And I had that.
How do you find a good mentor? This is, these are the questions that typically bubble up where people say, we tell people, oh, you have to network and you have to find a mentor. What is, from your perspective, what's the best way to find a mentor?
Well, that's difficult because you might not have much choice in the matter unless, if you have several different job offers. I would say that a future boss or supervisor that you can talk to like we're talking now. Who can be frank with you and you have a good rapport with, and trust your instincts, trust your gut feeling, because it's usually right.
Yeah, agreed. I always tell people that I feel like whenever you're interviewing for a job, it's very important to think about the fact that you are interviewing the company or the person just as much as they are interviewing you. And it's pretty much like dating. If your gut tells you to run, then run. But it has to be a mutual, agreement where everybody likes to work with the other person.
Yes. Science should be fun. And
Yes.
that's how I also deal with my trainees. Uh, I'm very interactive with my trainees. I tell them they can just pop into my office anytime I look like I could talk to them. And I pop into the lab as well, maybe too much for their comfort, but it's always in good spirit. And we generate a lot of ideas that way just by informal interactions. That's my style.
I think that's important, really having the opportunity to interact on a regular basis, because the questions that you're tackling, the questions that even people in the lab, in any lab are tackling, they're not yes or no questions. They're complex problems. And if we don't work together, we don't talk about it, it's very difficult to get to that answer.
And I also keep in touch with my trainees and try to help them with their careers later on. I tell them they're always welcome to contact me if they have any questions or need my intervention with something. So I get satisfaction from seeing their success.
And I think that's important when it comes to picking a lab, when picking a mentor and the supervisor to work with. All right. The other, well, we're getting almost on time. The other question I had, since we're talking about mentorship and about, fostering relationships, what would be your advice to junior scientists who would like to contribute to the field scientifically?
Challenge conventional thinking. Everybody says think out of the box, but it is true. You might come up with some perspective that the people who've been working in a field all along have just neglected. And also try to use orthogonal thinking towards things, take two unrelated approaches or ideas and say, is there any overlap of those things that I can exploit?
Yeah.
And another thing is one good piece of advice I got from one of my mentors, my postdoctoral mentor at Weizmann Institute in the Department of Organic Chemistry, told me always try to prove things as many ways as you can. Don't just rely on one piece of evidence and say, well, that probably means XYZ, because it might not mean XYZ.
Yeah.
And, always try to think about ways to prove your point that may not have been immediately apparent.
I think that's very important. That's a very important point, because at the end of the day, we're scientists, and if we can prove a hypothesis using different methodologies, that really enhances the conclusion and it supports much more strongly the conclusion. Any other advice before we move on to the next question I have for you?
Well, I know it's very challenging for scientists starting out, and they have to work typically for years before getting a permanent position. But my advice is hang in there if you can, because it's a very satisfying career. If that's what really is your natural inclination to do research on receptors, go for it.
Amazing. And obviously everybody should be working on GPCRs, because
Yes.
nothing else matters. I'm obviously exaggerating, but just by a tiny bit. Two last questions. We've talked a little bit in the beginning about the fact that you were at a very young age, exposed to chemistry and to the beauty of chemistry through your grandfather. Do you have any other aha moments that happened to you during your career that shaped your trajectory?
Well, finding out that I could attach the chains to my functionalized congeners and that I could even enhance the affinity. That was a huge moment. And I used that observation to test whether I had like an infinite space on the chain. So I think I made the very first biotin conjugates of GPCR ligands, and I took kind of a global approach, make a list of everything I can do with these chains. So I published a paper in 1987 where I had introduced fluorinated moieties for fluorine NMR. Of course, it took decades before people did fluorine NMR GPCRs, but it's in my paper, at least the ligands, not
Yeah.
not the NMR results. And I just looked at the global picture to say, how can I make use of this in every way possible? And I just tried all the variations, and that was making use of the eureka moment.
Amazing. Amazing. Any other aha moments or any other career defining moments? I think establishing the collaboration on the P2Y receptors sounded to me like something there that you found very exciting.
Yes. Well, I should mention the drugs we have in clinical trials,
Please.
which are all agonists of adenosine receptors. And that was a great lift, finding out that my compounds are safely administered to humans. And some of them have even proven efficacious in phase 2 and now phase 3 trials. So we're getting close. And they have very few side effects to speak of. So, for example, we have one compound that's in a second phase 3 trial for psoriasis. It's an A3 adenosine agonist that we made in the mid 90s. It's taken this long. And people might wonder, well, how does it work at NIH to get something on the clinical path? Do we do the clinical trials at NIH? No, the answer is no. We can't afford that, even though we're the government. We don't have blank checks in the government. I hate to
burst the bubble.
Yes.
So actually, tell us a little bit more about the story behind these compounds that ended up in these clinical trials. I think it's important to capture this because not a lot of our guests have the luxury of saying, I have all these compounds, like 4 compounds in clinical trials. And it's an interesting, I think, a goal that a lot of basic scientists can work towards.
Yes. Well, they were all synthesized in the course of making selective compounds for various adenosine receptors. And we just found their selectivity was very impressive. And then we started sending out the compounds to various collaborators. But even more important, we like to make our compounds available to the research community. Mm-hmm. And I have many dozens of compounds that you can buy. Most of our compounds now are designated MRS. So, for example, MRS 5698 is a highly selective A3 agonist, and you can buy it. And so we've learned a lot about our compounds from papers published where they just purchased our compounds. Mm-hmm. And it really stimulates the field to have these selective probes made available. And I'm happy to fulfill that function. I don't need to be a collaborator. I'm just happy that people are getting these things and using them, because it reinforces our work too. We learn about our compounds, and the 2 compounds that are most advanced in clinical trials were actually purchased as chemical probes by a small company. The company found they worked in their model, and then they came to me to try to explain it. And we had a 3-year official collaborative agreement with this company, and it helped them make progress towards initiating clinical trials. And that's how those two compounds got on the path. Now, we're not in any a privileged position with the company. Of course, as a government worker, I can't have any personal stock or in consultantships with the company. So these drugs that are in clinical trials are kind of like our children that have grown up and left the home and they have a life of their own. And sometimes they call back and let us know what's happening, but usually they just do their own thing. So we're happy to see the press releases about our own drugs.
That is still, I think that's really amazing to be able to, as you mentioned, seeing the press releases and knowing that you had something to do with that press release happening at all. You mentioned that you make these your compounds that you developed available to the general public, well, to the scientific community. Where can people purchase them?
Well, it's standard supply houses. I mean, you just look for pharmacological probes, neurochemicals. Well, Sigma, the giant, Sigma,
okay.
a lot of them. But I don't need to rattle off the names. You can any of our compounds, just do a Google search and you'll find typically a lot of companies will offer it.
Got it. Yeah, I was just wondering if there was a specific, space or a specific company that had it, or sometimes there are nonprofits that also are like, I'm thinking here about AdGene as the plasmid repository.
Right, understood. Well, there is something like that NIH sponsors. There's the NIMH Drug Repository and Synthesis Program, and some of our compounds are in that. So you can get them for free with justification from the NIH Drug Synthesis Program.
That's amazing. I think that's also very important for the audience to know, if in case anyone is working on the adenosine receptors and any other receptors when it comes to compounds, to know about these resources.
Right.
Ken, last but not least, if and when you have job openings in your team, where can people find you?
Well, I'm constantly getting applicants by email. That's the best way to give it a try. And if I happen to have a postdoctoral opening at the time, then the person fits in to what I'm looking for, then I will write back with an encouraging email. I don't. Sometimes I advertise on NIH websites. If you search through NIH, you can find a listing, not only of my lab, but many labs, mostly are biological labs, but a lot are working on GPCRs. For more junior positions, we have summer students and post-baccalaureate students who come for a year or two between undergraduate and graduate school. And those are applied to through the Office of Training of NIH. It's called OITE. Just Google search OITE, load your credentials and your professor's recommendations. But I have to emphasize, you have to, as an applicant, you have to reach out to prospective labs. Don't just think because you have submitted your data that somebody's going to hit upon your application and choose you as a candidate. You have to solicit from the labs that you're most interested in, which you can find on the web. So that's how we find postbacks principally.
That's amazing. Yeah, and I think it's important. I think that goes back to the advice that you were giving to the junior scientists. It's about being proactive, just like you as you were before getting tenure, and throughout the conversation. Yeah,
I should add that we also concentrate on maintaining diversity in our working groups.
That's great. And that's also very important, I think, to have that diversity. I think the more diverse the group, the more diverse the perspectives are, and that enriches definitely the scientific interactions. Great. Well, thank you so much for your time, Ken. I really appreciated your time. What a great story. I think you're our first chemist who actually had a family member inspire them to go into chemistry. I think we've had previous guests. I think Brian Triquette's background is also in chemistry, but he had a very different path. And I like to talk to various people. I'm trying to bring in more guests who don't have the traditional, what I would call the traditional GPCR background, working in live cells, in HEK cells, and doing one type of assays. I think it's great, but enriching the field with the ability of making different compounds, it's just phenomenal.
Thank you so much. I've enjoyed our discussion very much.
Same here. Thank you, Ken. Thank you for listening to this Dr. GPCR podcast episode. We would like to thank our guest, our Dr. GPCR team members, Attila, Ines, Monse, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with us and with our partners directly in the ecosystem, so make sure that you join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.